This meeting isn't always perfect, but it's always great. I really love coming here because it was the place I really got a first exposure to endometriosis. So what I'm going to talk about today are some tools that we've been trying to bring into the endometriosis research community after my coming to this meeting for many years and interacting with surgeons here. And I want to especially call out Tamer and Padma for starting this meeting and Lona for helping us all stay engaged across many different kinds of disciplines. I came into the topic I'm going to talk about sort of a back way. My research is in tissue engineering, and this is from an article that Science Magazine asked me and Gail Naughton to write in the early 2000s, where we describe regenerative medicine applications. But at the end of the article, we gave the perspective that the field was really going to move toward building in vitro models of human tissues for better drug development. And this is a picture of a liver model that we've been developing in our lab that's now been commercialized and used by a lot of pharmaceutical companies in drug development. So over the past decade or so, my lab, but the whole field in bioengineering, has moved more toward these so-called organs on chips to use in drug development. And what I'm showing here on this slide are a few examples from the field, work of my colleague Roger Kam in building microvascular networks. You can see endothelial cells in a tiny gel in a microfluidic device. This is a picture of the device. Cancer cells and immune cells moving through these patent vessels in this little device. Other examples from people in the field really trying to do tissue engineering now with devices to let you do fluid flow and so on and so forth. So this is a huge area of investment by the federal government, and DARPA, the Defense Advanced Research Projects Agency, and NIH together are spending about $100 million in a 5-year microphysiological systems program. I'm one of the primary investigators, so I have over $30 million during this time period to do research on how do we build mimics of human physiology for drug development. Okay, so it's really hard to get funding for endometriosis. Everyone knows that, but there are ways to get funding that you can bootstrap toward endometriosis, and we have done that. My DARPA program manager has let us do that. So this is a drug development pipeline, and in the context of endometriosis, I think about this all the time because we know it's been extremely difficult to get drug companies to develop things that are not hormonal regulators or plain anti-inflammatories. So the part of it that we brought in to this whole program and is now very visible at NIH, I go there and I talk about endometriosis, and I'm going to give you a short version of this talk. So what we bring in, normally you have patient population going to target identification, and we've had some beautiful talk today from Ozgi Fazlabas about mechanisms of endometriosis, regulation of gene transcription and so on, but we also now want to think about expanding mechanisms to stratify patients. So the organs on chips started out, most people thinking of using them for toxicology, but we have been focusing on trying to bring them in to earlier stage drug development. And so our goal here is to start with clinical data. What is the patient population? And a message from today is that we think that there may be many different subgroups of endometriosis patients. You can see Padma here, my niece here. All of them have symptoms, but they may have underlying different mechanisms of disease ultimately, and how can we start to tease that out? So we're using this approach, thinking about stratification, to design in vitro systems for studying disease with a primary focus on endometriosis. And now, I've given talks here before where I've used this slide, but it's just to orient us that we stratify patients all the time in cancer. Cancer has known genetic mutations. Endometriosis, no known somatic genetic mutations, though there may be epigenetic modifications that are regulating the disease. So right now, if you get breast cancer, you're immediately stratified by 3 molecular markers used around the world. Those have value for prognosis and for therapy, and there's targeted therapies for all of them. Can we move toward that in endometriosis? Now, when I started working with surgeons, I found it very interesting. It's repeated over and over again that the staging of the disease doesn't really predict prognosis and therapy all that well. Patients with a little bit of disease can have a lot of pain and vice versa. And one of the things that's come up today, and as I'm starting to become more attuned to it, is maybe some of these patients who have minimal disease and a lot of pain also have other diseases like adenomyosis that need to be attended to. So where does this take us? I'm going to do a short recap of what we published on our stratification and set the stage for tools we're bringing in the community and working with some people here and elsewhere to try to understand the crosstalk between the endometrium, the peritoneal lining, and peritoneal fluid in inflammation and endometriosis. So we've, again, heard beautiful from Ozgi this morning about this crosstalk as studied in baboons. We want to bring new tools that will let us take patient samples and start to study it in vitro in a defined way. Now, we got started in this by focusing on peritoneal fluid. Peritoneal fluid bathes the peritoneal lining. It's where a lot of inflammation is known to be taking place. And if we, we start here thinking about the communication networks between these different cell types. So a level up from what Ozgi talked about. So you've got genetic and RNA, and then you have the proteins and protein network states. So can we start to build models that let us probe the networks of communication? Okay, so this is a simple version: immune cells, stromal cells, epithelial cells. Here's now starting to put layers of mechanisms of networks of things happening. So immune cells talking to each other and to the endometrial cells through cytokines, but also all kind of invasion networks regulated by proteases, regulated by growth factors. Networks of things going on. Can we as engineers start to build a model of these networks, communication flow between cells and within cells, to unravel the disease? And most importantly, can we use these networks to maybe classify patients or start classifying them according to networks that may be most dangerous in them? So we took an approach, and this has been published with Keith Isaacson taking the peritoneal fluid, very easy to get, took the cells out and analyzed some of these networks. And briefly recapping work that we had published, we asked if in groups of patients, we had a reasonable sized patient population, all of these samples, we asked, are there inflammatory cytokines that change at the same time in groups of patients? And we did a completely unsupervised kind of math that Netflix uses to tell you what movies to watch. We found a group of 13 cytokines that changed in concert in these patients. And importantly, what does that mean? Who cares? It contained patients from all stages of the disease according to the surgical classification, and there were about a third of the patients with this particular signature. There was a trend toward enhanced pain. We've subsequently done an initial study with Maurizio Abreu, and amazingly were able to replicate the analysis identically. We had a completely different set of patient demographics because he has a different kind of practice, but we came up to very similar conclusions. In fact, the same 5 cytokines emerged when we did a meta analysis, and that manuscript is in preparation, and it's my fault that it's not been published a year ago. Postdoc left the lab, and I'm very slow writer. What we found in this study, though, is we identified by reverse engineering a macrophage phenotype responsible. We went back, took macrophages from patients, their peritoneal fluid, and showed they were making those cytokines, control patients didn't. And in studying these macrophages in culture, we identified Jun kinase, which had not previously been identified as a major regulator of the production of these cytokines. And so that was very interesting. Hold that thought about Jun kinase. We then wanted to study invasion networks. And again, this is a picture from Maurizio surgery. I went to see him on vacation one year, and he entertained me. He took me in the OR. It was amazing to see the whole setup he had there, and he was doing a bowel resection of this patient that day. But it made us really think about invasion networks. Now, you don't need to think about the busyness of this slide. This is simply the other part of it. So we talked about immune networks, and engineers like to conceptualize things. Information flow. So we have a cell that's getting cues from growth factors and cytokines. It goes through signaling networks, and there's proteases on the surface of the cell, enzymes that are shedding things, the receptors, the growth factors, and so on, and then the cell does something, it invades. Okay, so this is an engineering conceptualization. We can put math on all of those steps. We can make a network where we put rates and numbers and so on the different steps. And I'm not going to try to teach you math today, but what we do is we build a model that we go in and we can measure all these things, and it's expensive and time consuming to measure these, but it helps us start to get at mechanism. So again, you don't need to know the details of this, other than we can start to think about phenomena in engineering ways. Now, we had to invent some new ways to measure protease activity and collaborate with other people. And you're familiar maybe with seeing heat maps. This is just a whole bunch of different molecules in the peritoneal fluid. This is a whole bunch of different proteases, cell surface and otherwise. We're looking at their activity, their concentrations. The point is we can get a lot of data, again, out of the peritoneal fluid, and we can come back and we identified some mechanisms of invasion that again involve Jun kinase, but we're really trying to ask the question, can these networks help us classify or stratify patients? With the protease invasion, we only were able to do a pilot investigation because the analysis turned out to be really hard. We did 9 patients, and indeed, we found 2 groups of patients separated by some protease activities. And again, I'm not going to go into the math that is the discriminant PLSR modeling, but these are very provocative early data suggesting there may be value in taking these mechanism-based approaches. So early provocative data, but not definitive. And we want to expand these with more studies. What's very intriguing, there was a Jun kinase inhibitor trial going on in Europe by Preglem. The trial was never published. We've heard, not definitively, that it was some patients responded really well, others didn't, so the average trial couldn't go. Now, what we've been doing more recently is saying we would love to build models of how these cells are all interacting in culture so that we can actually study patient samples where we recreate these tissues in culture. So this has been a huge focus lately, and we are working with Ozgi Fazlabas and Kevin Osteen because we got a grant to try to transfer these models out to the community. So many people, if you do cell culture, you probably use Matrigel. It's an extract of basement membrane that comes from growing a mouse tumor. You grow the tumor out under, you extract this goo out. You can buy it now, but it's still not a reagent. So we set out to say, can we build a completely synthetic extracellular matrix that will let us rebuild the endometrium or the peritoneal line? The salient features are epithelial cells, stromal cells, immune cells. We will do vascularization, but not yet. And moreover, can we dissolve it so we can get cells and all the molecules out so that we can study them? So we want to be able to build these tissues, 3D models, and perturb them. And we've come a long way toward doing that. So the basic way we start is we take a completely synthetic polymer called polyethylene oxide. It's used very widely in biomaterials, thousands of labs, well, hundreds anyway. Hundreds. You combine 2 different things together. They cross-react. They're very gentle. You can put cells in it, get a very clear gel. Shown here, it looks like a contact lens. So what we've done, we have done a lot of fancy engineering of this, where we put goodies in there that will bind to the integrin. So this is showing a cell membrane, the adhesion receptors that bind to matrix naturally, bind to a little teeny tiny mimic of a matrix protein of fibronectin. They bind to a little mimic, but then we also have things that help capture things that cells secrete to make their little nests. So this is a molecule called fibronectin. It's a natural part of your matrix, and it's growing there. So we can now build the endometrium. Lona says I have a minute and a half, and so we're really at the end. We can build the endometrium, beautiful epithelium, and we look beautiful stromal cells making matrix. So this is all grown in culture from human endometrial cells. We can do decidualization over a month, giving hormones to cause proteins that are normally involved in decidualization to be secreted. And moreover, we're now incorporating immune cells. So we can put immune cells in this and they migrate around and differentiate into the normal cells, and we have been successful in breaking the matrix down and measuring cytokines within that matrix. So now we've got all these tools to really study the signaling networks in the matrix. And finally, we are now starting to put these on our organs on chips. I've shown you everything in culture. DARPA program, we built this huge platform with little microfluidic pumps to connect an endometrium model with a gut and a liver and something else, so we can actually start to look at endometrium interacting with other things. So our goal is to really bring these kind of tools into drug development for endometriosis. This is an example. We don't have endometrium, but this is gut, liver. When they interact together through these pumps, we completely rewire the function of the liver in a beautiful way. So we're really moving toward in vitro physiological human on a chip. And what I hope I've convinced you is that these tools may be useful in the endometriosis community for studying mechanism, and that's why we're trying to get them to Ozgi's lab and to Kevin's lab and others.